So I'm Kelly Stratton. I'm a statistician and the integrated research platform lead for data transformations in EMSL. Satish, I'll let you go ahead and introduce yourself briefly, and then you can go to the next slide.
Hi, everyone. I'm Satish Kara. I'm the integrated research platform lead for systems modeling at EMSL. Are you able to see the slides?
Yes, we can see them.
Okay.
Can you advance it one, please?
Oh.
Thanks. So to start off, we just wanted to give an overview of BER research, as I'm sure you're all aware, and how Satish and I and our roles fit into that, and what that can bring for EMSL users. So the BER research community has two divisions, as we know: biological systems sciences and earth and environmental system sciences. And EMSL is structured to reflect this, but has one key difference. So there's one science area that corresponds directly to each of those BER areas, but there's an additional third one that is computing, analytics, and modeling. And this was created in the more recent history of EMSL to really provide us the ability to focus on the computational aspects that will drive science forward and complement and collaborate with these two other domains. So at EMSL, each of the science areas is further subdivided into integrated research platforms. And next slide, please, Satish. So as we introduced ourselves, Satish and I are the IRP leads for two of these integrated research platforms. We wanted to also put a face to the name of Jay Bardhan, who is the science area lead for computing, analytics, and modeling. And we have a third IRP that's still under development called computing platforms. And many of the intended purposes of this are currently being handled through Satish's IRP, as well as the computing and operations team at EMSL. Next slide, please. So to give you a little bit more flavor of what each of these IRPs helps steward, we just wanted to briefly say that the systems modeling IRP really builds on EMSL's history of leadership in simulation-based science. So this includes modeling approaches that might be translational, where we are bringing externally developed capabilities in-house and making them easily available for EMSL users. It includes data-driven modeling efforts and also embedding all of these modeling capabilities into EMSL workflows. For data transformations, the purpose is really to maximize the value of EMSL generated data for our users. So this includes things that help streamline or enable automation from a computational perspective. It involves how we package and handle the data products that are generated, and includes things like statistical analysis, data integration, visualization, and so on. And then the computing platforms IRP is going to center on making sure that the EMSL user community can fully leverage both the hardware and the software that are supported, developed, integrated into the different workflows available at EMSL. Next slide, please. Thank you. So each of the EMSL science areas has a strategic objective that they steward, and for CAM, that is modeling and data sciences. And this really intersects nicely with the topic of this workshop, in that one of the main areas of focus is computational imaging. We also focus on computational mass spectrometry, advanced computing technologies, as well as MODEX-driven simulations. And this, if you're not familiar with the term MODEX, is simply the interplay between experiments, modeling, and driving the next round of experimentation. Next slide, please. So to give you a flavor of how the data transformations IRP can interface with different steps along a project or experiment life cycle, I wanted to sort of walk through this diagram with you. And this really begins with experimental design. So depending on the complexity of the experiment and the types of analytics that are of interest, this can end up being extremely collaborative. It can include the EMSL users, it can include experimental scientists here at EMSL, the instrument scientists, as well as someone with the statistical knowledge to make sure that data that are generated are really going to be able to help answer the research questions that are of interest. Once this has been solidified, of course, the experiment can be performed and that will generate data. And while there are a lot of externally available and proprietary software for doing things like molecular identification and annotations, there are also some tools that have been developed here at EMSL that can facilitate doing this in a very streamlined or even automated way. So those are highlighted here. In terms of the statistical analyses, this is really another area where data transformations comes into play. Depending on the familiarity of a researcher with the type of data that they're generating, they may or may not need someone additional to help on this front. Especially like we saw from Alok's presentation, where they're generating 4 different types of data streams. Things can start to really get more complex, especially when you're trying to pull in additional information like they've done. We saw some pathway information, right, be pulled in on some of the figures that she showed. And I appreciated that because we often go through, at least in my experience, you do your analysis, and then you start to realize that you need more information to really interpret your data. So that's why we've built into this figure these smaller feedback loops, right? You might pull in some information, make a visualization, and realize I don't know enough to really understand what's going on. I can go pull in pathway information or something like that. And then, of course, at the end of our project or experiment, we've hopefully made some scientific conclusion that really drives forward our next proposal or our next experiment to keep furthering that knowledge. So Satish, with that, I will go ahead and turn it over to you.
Thank you, Kelly. So the systems modeling integrated research platform aims to accelerate scientific discovery in the BER space by developing advanced modeling approaches together with high-performance computing and machine learning and AI, going from molecular scale to microbial organic matter to soil, all the way to ecosystem scales, and also building essentially algorithms and techniques to bridge all these scales and put them together. So at EMSL, we've been investing in developing approaches built on machine learning and AI. You've seen several talks yesterday and some today that essentially, together with our in-house high-performance computing infrastructure, we have a cluster called Tahoma, and with accelerated computing architectures like GPUs, to essentially mine information from multiple modes of data coming from our instruments. Together with physics-based models, we are enabling the ModeX paradigm that Kelly talked about, essentially, which is the model experiment exchange, right? Paradigm for the BER. So I will highlight a few examples in the chemical imaging space, the next set of slides that we've been working on and we've been investing in. So the first example is the integration of 2D chemical image data with 3D structural data. In this case, I'm showing an example of image chemical data coming from SEM EDX, and then the structural data coming from XCD. And then this co-registered data using machine learning techniques is then used to create these 3-dimensional meshes, essentially with pore structure and chemical information embedded with them. And those are then used to perform pore scale modeling, essentially to do flow and reactive transport modeling, and then to calculate parameters that are then used to upscale and then move them to higher scales, such as the watershed scale or even higher. Also, algorithms on pore structural information from, for instance, XCT are being used to perform or to build lab-on-chip designs for performing controlled experiments to understand aspects such as nutrient and chemical transport. This is the pore-to-chip on the top that I'm showing, the pore-to-chip technology and the capability that Arunima actually highlighted nicely yesterday. We are very excited about these integration between the modeling and our algorithms on the CAM side of things with the experiments. And then on the bottom, what you're seeing is our vision for the next steps are taking the data from these micro models. The data is then used to validate our pore scale models. And these models are then used to inform higher scales. So, like I said, using upscaling techniques. You've seen some of the approaches that Maruti discussed about in terms of upscaling. And so we are really excited about this whole loop of MODEX that we are enabling for the BER community. In addition, in the space of mass spec imaging, we are developing approaches for semi-automated co-registration and image segmentation using essentially techniques from statistics and machine learning. These are to identify similarities and quantify comparisons, and also to identify spatial clusterings in the images. And then also at the heart of our EMSL infrastructure and our data service ecosystem is our commitment to serving data on demand. And this is crafted through the lens of user-centric design. So the approach is not just about technology, but also it's about the people. So it's about understanding the real world context in which our scientists and researchers are operating, and essentially tailoring our systems to meet those needs, meet the needs of our users. So our infrastructure is built on the principles of primarily collaboration and sharing, breaking down essentially the silos that often happens in the research communities, and then fostering a community where data is not just available, but also accessible in the most practical sense. And so towards that, we've been investing in making data accessible via web-based APIs, and providing access to the biological and environmental science communities, and enabling the integration of our data with other research organizations. And then this is, again, with a standardized language to ensure clear data communication and transition from between various institutes. Also, the last, essentially the paradigm shift at EMSL that comes in the form of essentially instead of having focus on individual projects and instruments, but in terms of enabling data-centric portal that is intuitive, user-friendly, and powerful with the orchestration of both biological, biogeochemical, and environmental information. And this is in the form of searchable, filterable, and has the ability to integrate with other data services, like I mentioned, through our APIs. And this is a part of our new Science Central platform, which we are excited to have introduced this year. And so putting all this together, like I said, we're building on this new EMSL Science Central platform with the vision to seamlessly integrate all the data coming from the laboratory instruments, the analytical results, and then enabling downstream data analysis, exploration, and integration with models. And so I've highlighted the URL here. If you're interested in checking out, please go and see the new EMSL Science Central portal. And that's all for today. That's thank you for your attention, and please reach out to us if you're interested in collaborating or learning more about computing analytics and modeling at EMSL and how we can help accelerate your science.